Okay. So whatever we learned about correlations and noise correlations, etc., we are going to today see how they are used in an actual, recording, experiment, and what can we learn about, how the brain works in some ways by looking at the responses. So here's one question. Suppose you've got two neurons. Okay, so which like different orientations. So what you are doing here in this study is that you are recording from a bunch of neurons simultaneously from the primary visual cortex. Okay, so tuning curves of two V1 neurons here are shown in these two curves. Tuning curve just means that as I change the orientation, how is the response going to look like? Okay, so that is the tuning or the preference of the neuron. So one neuron likes 0 degrees, about 0 degrees, the other neuron likes 45 degrees. Okay, so they have slightly different tuning preferences. Now for every orientation, so suppose I have presented a stimulus at 0 degrees. Okay, so I have given you two neurons, so you have data, but for us only a single orientation. Right, so 76 trials that you have in your data set are all for a particular orientation. If I change the orientation, I can do this experiment after changing the orientation to some other value as well. Then I could actually generate tuning functions like these. In fact, we have done it for different orientations. I just have given you data for one particular orientation. And we computed the spike count correlation for that orientation, right? So remember, let me just quickly go back to, yeah, so this thing here. This is spike count correlation. Number of spikes from neuron 1 versus number of spikes recorded from neuron 2 when the stimulus was the same. These are 76 dots, each corresponding for one trial, and this is the correlation, spike count correlation, which in this case is 0.2 during the stimulus period. Okay, now the question is this spike count correlation going to change if I change the orientation? If I had done the experiment for a different orientation of the stimulus, which may or may not have excited these neurons as much, would I expect the spike count correlation to change? Okay, everybody gets the question, right? So that's what is being answered here. I can look at any particular orientation. So let's say orientation B, which is minus 22.5 degrees. And then one neuron is firing an average of these many spikes. The other neuron is firing an average of these many spikes. But these are averages of lots of trials. So suppose they did the experiment 50 times or 100 times, whatever. This is the mean of that. But I could make a scatter plot of the response for each trial for cell 1 versus cell 2, just like what we have done previously for those 76 trials. And then the spike count correlation is just the correlation between the two. And in this case, it's about 0.26 for orientation B. And then the question is that if we choose one orientation that, is like the preferred for both, if the tuning curves are close to each other, is the correlation is going to be higher than when the orientation is suboptimal for both. This, of course, question only makes sense if the tuning preferences are more or less similar. And I actually would have thought that it would matter. The orientation that neither of these neurons like, let's say this one, 90 degrees, neither of them like, would produce, weaker spike count correlation than, let's say, 0 degrees. But that is not the case. Here you see, if you look at different orientations, the spike count correlation is about the same. It doesn't depend on which particular orientation the stimulus is presented at. Okay, and this is something that you can only do an experiment and find out. This is the summary of this. If you look at the best orientation, the average, this is over the population. The average correlation is about 0.2. This is the spike count correlation. And it doesn't really change. It's about 0.2 even for worst orientation. This is the average of about 100 neurons. Okay, so this is cool. I mean, this is the, 100 pairs. We found something about correlations and its dependence on stimulus, the stimulus that is presented. And turns out the stimulus orientation doesn't matter. Okay. So this is one thing that we learned. The other question would have been looking at the CCGs, the spike count, not the spike count correlation, but cross correlograms, the ones that we discussed here, these magenta traces in the previous class. Everybody remembers that, right? I hope everybody remembers that. So this is now here, this is cross correlation, doesn't show much of a peak, but here in their data, they have averaged over a lot of, no, sorry, this one is not average. This is just a single pair. But here you do see a bit of a peak at 0 degrees. Okay, so the cross correlation does show a bit of a peak for the best orientation, but it sort of goes away for the worst orientation. So now you might ask, hey, how is it the case that I'm seeing a peak here, but the spike count correlations aren't changing? How come the spike count correlations are about the same, but the cross correlation function looks different? So anybody has an answer to this? This is related to the relationship between spike count correlations and the cross correlogram functions that I had briefly discussed in the previous class in response to a question that was asked by one student. Any thoughts? Anyone? You guys can hear my presentation, right? I'm not.
Yes, sir.
Okay. So what do you guys think? The cross correlation function is showing a peak, but the spike count correlations remain the same. How is that possible? Okay, so I'll give you the answer here. The spike count correlation is the average, so when I look at these dots, the responses, these are average responses over some time interval. In my previous thing here, when I was computing this was averaged over 256 milliseconds. Okay, and this one, this other measure, the spike count correlation, sorry, not the spike count correlation, the cross correlogram, this is a function of time here. Now in Smith and Co, this paper, they show that to a large extent, the spike count correlation happens to be the integral of this over some interval. Okay, so if you integrate this function over plus minus 200 millisecond, you will tend to get something that looks like the spike count correlation. So even though the peak is different, it's a change only at a very small region. So the integral is not large because the difference is over very short time interval. It's this little bit that you are seeing here. This thing dominates. This little, increase that you see, it will become stronger when I show you the correlation contrast plots in the coming slides. So this difference at a single value at 0 time doesn't contribute much to the spike count correlation. Okay, and this is why even though at a single value at 0 millisecond the peaks are different, the spike count correlations don't vary much. Okay, and here this is quantified. You show the peak height versus for the best orientation versus ineffective orientation, and the peak height is different. So there is a difference in the way these neurons are firing when you change the orientation. But the width is about the same. The width doesn't vary. And these guys even did this integration over varying windows. So you change the window of integration. The best orientation is shown here in light gray. The best worst orientation is dark. So if you had integrated or computed the spike count correlation over small intervals like 1 or to 10 milliseconds, you would see the difference. But as you increase the integration window, this difference fades away. So if you are integrating over 500 milliseconds or more, then you don't see a difference in the spike count correlation. But if you had integrated over a smaller integration window, then you would have seen a difference, because then this peak here is not insignificant.
Sir,
Yeah.
could you please just define what is peak width and peak height? I couldn't get it.
Well, the peak height, as the name suggests, is the height of this peak.
Okay.
And width is,
Oh,
on this function, what the width is. That's it. It's nothing more than that. Peak width and peak height. So I'll show you another type of stimulus variation, and then it will make much more sense. So let's just look at that. Now what I'm doing is I'm changing the contrast of the stimulus. Contrast just means what's the difference in the luminance between the black and white portions of the stimulus. So this is a highly visible stimulus and this is a barely visible stimulus. Okay, so this is what means by a high contrast stimulus and a low contrast stimulus. So in this paper, there are two types of modulations. Either you keep the contrast the same, and then change the orientation. So this is horizontal lines here. You can make it vertical or 45 degrees, and that's the manipulation that we have seen here. These are the different directions. Or you hold the orientation the same and change the contrast. And if you change the contrast, see something very interesting that is happening. What you see here is that at high contrast, you see this nice peak here. As you reduce the contrast, this peak goes away. But on the other hand, this bump here starts to appear, this low, this sort of, larger time duration, but slowly increasing bumps. This is time axis here, and that sort of disappears at high contrast. Okay, so this paper is actually hard to read. I mean, you have to spend maybe a couple of hours just trying to read and try to this thing to sink in. But what I want you guys to appreciate, that CCG and spike count correlations, they are related in some ways. But as you change the stimulus, the neuronal responses change in very different ways. Okay, there are differences in the way they respond, And those are captured in different aspects of the CCG and spike count correlations. So here, as I'm increasing the stimulus contrast, the synchrony is increasing. Synchrony is the zero, this peak at 0. That's the synchrony. So stimulus contrast increases synchrony, but the overall integral, the spike count correlation actually decreases, because this area under this curve is much more than the area under this. And that is also shown here, that at worst contrast, the spike count correlation is actually higher than at lower contrast, at higher contrast. It's 0.25 here and it's about 0.4 here. Okay, and it's quantified here as well. If you are integrated over a small window, this is the synchrony aspect. Then the best contrast, shown here in gray, has higher area under the curve than this, but that it changes. The lower contrast, because it has this thicker sort of slow, the area under this curve is more, you see that here more than this one as you increase the integration window. Okay, so manipulation of contrast and manipulation of orientation has very different effects. And a simple measure like spike count correlation and CCG is able to capture all of this. It's quite interesting if you think about it. The math here is not very involved. The math here is easy. I'm just looking at correlations. And the CCG, which is normalized in some way, it's also not that hard. We did that in the last class. We're not talking about very advanced math here. But we are looking here at the way the brain responds and codes the contrast of the stimulus and the orientation of the stimulus, and there are intricacies there. And as neuroscientists, this is what we try to understand and pull out from the data.
Hello.
Yeah. Yes.
Yeah, I cannot hear Supratim. Is there anyone else who is there?
No, we're able to. I mean, at least I'm able to hear.
We are able to hear.
Okay. So it's a problem with you at your end.
Okay.
Okay. So, well, it will be made. All the lectures are online, on the course web page. So if you get cut, you can always, look at that later. Okay. Okay, so I'm done with this part. I won't spend more time on this. I know that I have gone through this in very quick time. So if you are confused, you have every right to be confused. As I said, this usually takes about 2 classes for us to go through very slowly and all that. And unless you read the paper up front, there is just too much information to handle. Okay, this is another interesting thing, figure 12. I won't talk about it, but I've given you this as an assignment. So you'll have to really read the paper and try to understand what this plot says and write that as your answer. So please do so. Everything will make sense. This RCCG and spike count correlations and how they are actually important in our understanding of how an ensemble of neurons together code information. This whole picture will become very clear if you read this paper, but it will take you a while to read it. It's not, if you're reading papers for the first time, it will take you a while to read it. So please do so and then go through these slides, and then it will make more sense. Okay, so with that, we are done with the spike module, and now we will start with the LFP module.
So
yes,
I had a couple of questions based on the paper.
huh.
So there are three terms they've used throughout the paper. It's called the best orientation, the worst orientation, and something called the best compromise orientation.
Okay, yeah.
So, my understanding is that from figure 1, where they've made those two tuning curves, the best orientation is perhaps the intersection of the curves.
Well, so best orientation is defined per neuron. Okay, so every neuron has a best orientation, which is the orientation at which the firing rate is maximum. So for neuron 1, the best orientation is minus 22.5. For the second neuron is 45 degrees.
Okay.
Worst orientation similarly are different. These two terms are defined per neuron.
Okay.
Now, unfortunately, all the analysis is done for pairs.
Okay.
That is where the compromised term comes in. The best compromised orientation is the orientation that elicits the best response for the pair.
Okay.
So in that case, here it would be about this middle one here.
So the intersection.
Intersection.
Okay.
And it would barely make sense if the tuning curves are too far apart.
Right. Okay.
Okay, and then the question is the noise correlation, which is the average of these values, dependent on the separation between the tuning curves? And that actually is figure 12. The signal correlation is a measure of how far the tuning curves are. Okay, and turns out, so that's actually the homework question also. So the noise correlation does depend on signal correlation, which means that the values that you are seeing here, they don't depend on which particular orientation you are presenting, but they do depend on whether these two tuning curves are close to each other or not. The closer these tuning curves are to each other, the higher is the noise correlation.
Okay.
Okay, so you have to really read this paper and to make this clearer.
Yes.
But I'll leave it as an assignment. Any other questions before we move on? All right, then we move on to local field potentials. Okay, so let's get going on this. So the references for this part, are just a couple of review papers, very nice review papers. So please read this, and this is sufficient for you. So I'm basically going to spend a fair bit of time talking about where this signal is coming from, what are the contributors, and what is the spatial spread, or how many neurons contribute to the signal. If there is time, I will talk about relationship with spikes. Otherwise, we will cover this later also once we have learned the techniques for this module. Okay, so here's the experiment. Well, it's the same experiment that we had talked about earlier. We take a microelectrode. It's a very thin microelectrode, and we push it inside the brain of a monkey or a rat, or whatever, some animal, and this electrode sits very close to a neuron that I'm recording from. Okay, so in the first module, when we were talking about spikes and I was showing you the picture of an action potential, was the recording set up like this, or was it different?
It was different.
How was it different?
Like the electrode was inside the cell.
Electrode was inside the cell, right?
Yeah.
Very good. What was the potential outside the cell?
So the potential outside the cell, it was positive.
And with respect to the inside, right?
Yes, with
So
respect.
the action potential, when we all talk about potential, is always with respect to some other potential. It's the potential difference that we talk about. So when we were looking at the intracellular potential, it was with respect to the outside, right? The potential was minus 70 with respect to the outside, right? So In this case, I have got a single microelectrode here. Where is the other electrode? What is the potential that I am measuring with respect to?
So the other electrode might be grounded, like zero potential.
Well, so yes, you define something as zero. I mean, there is absolute terms, there's nothing, fine. Yeah. So you measure the potential with respect to another electrode, which is somewhere far away from this electrode. And I'm saying that that potential is, this other potential is zero. So that is what my ground electrode is, as you rightly point out. And I measure the potential with respect to that. Okay, so now based on everything that I told you in the previous module, this is not a straightforward question. I'm sort of trying to trick you guys, so don't get tricked and think this through. So based on the previous module, when we were talking about the biophysics, what do you think was the potential at any location outside the cell? Okay, I see silence here. So I'll give you the answer. The potential outside the cell was always assumed to be zero. Okay, so inside the cell is different from outside. But in our description, we never actually talked about any potential difference outside. It didn't matter where I kept the electrode. It could have been here or there or there or there. It wouldn't matter. As long as it's outside the cell, we would assume it to be zero. And actually that is true for, as a first approximation, it is indeed true, because the potential outside the cell doesn't change much if you change the electrode as compared to inside. Now, if I show you the signal outside the cell, this is the extracellular potential as a function of time. This is real recording. This is time and this is voltage. Look at the range here. It's only plus minus 200 microvolts. The extracellular potential is barely more than a millivolt. It's very small. What was the potential difference inside across the membrane when during an action potential? How much did it change? Anyone?
In terms of millivolts.
It was millivolts, right? Minus 60 to plus 40 it went. So there was a change of about 80 millivolts or so. But if you that was when one electrode was inside and the ground was outside. Here we have a different situation. There's one electrode that is outside with respect to another electrode that is further outside, somewhere else in the cortex. In that case, the potential difference is only of an order of a millivolt or less. So as a first approximation, assuming that it's a constant potential is not a bad approximation. But this is the only potential that we have access to when we are recording from outside the cell. Turns out if you try to insert this cell inside this neuron, you keep pushing the electrode until it pierces the neuron will die. These electrodes aren't fine enough for us to be able to, preserve the neuron without killing it. And you have other type of techniques, these glass pipettes that are patched and then you apply suction to open, it's a different technique altogether to record intracellular potentials. These electrodes, the microelectrode recordings that we are looking at, these are actually, these are only for extracellular potentials. Okay. So the raw signal looks like this, and it's only a few microvolts, okay, not much. if I look at the high frequency content of this is the signal I get. So remember in the previous module, I was actually telling you that, you present a stimulus and you get action potentials. We usually don't do those recordings intracellularly, even though people have done it and it is possible. It's done through extracellular recordings, and you get traces like these, and these vertical ticks correspond to the times when when the neuron that your electrode is very close to is firing an action potential. So you can put a threshold. You can say it's minus 65 microvolts or so. And anytime it crosses that, or minus minus 50 or whatever, I'm going to count that as a spike. And these are those spikes. This is based on some threshold crossings. And these are the spike trends that we used in the previous module. These are the ticks. This is the time series that we analyzed and worked on. Okay. However, if you instead you take the raw signal and look at the low frequency component, the slow wave on which these spikes are riding, that is called the local field potential. Okay, so it is a signal that is recorded from the same microelectrode that also gives you spikes. It's the low frequency component of the raw signal. So it's obtained by low pass filtering the raw signal. And we will talk about filtering and low pass and all of that for, the next 10, 15 classes. So, you'll get to know what all of that means. It is a continuous signal. So as a function of time, you get a value at each time point, unlike spikes, which were a discrete, it was a time series of events. All I gave you was a bunch of spike times. Local field potential is voltage as a function of time. And it is a small value as compared to the intracellular potential. And today's class, we are going to talk about local fields. But actually, if you look at brain signals, other than action potentials, the rest of the signals, at least the electrical signals, have a very similar sort of structure. They are all time series, voltage as a function of time. People record signals by putting different types of electrodes. So electrodes can be placed on the scalp, which is called EEG, or it can be placed on the surface of the brain, which is called ECOG or electrocorticogram, or you can insert microelectrodes and record extracellular potential like this, and that is called local field potential. They actually have a similar structure in many ways. And that we are going to try to understand what this signal, where this signal is coming from. Okay, and these are all electrical signals. There are also magnetic signals that you can get. So fMRI, MEG, these are other type of signals that we are not going to consider because of lack of time. But together, they all tell you something about how a neuron or a group of neurons, what kind of information they get and transmit. We will see what this is. Okay, so where is this signal coming from? Why am I getting this fluctuation? More importantly, is it just spiking activity of a group of neurons, or is there more to it? Turns out this signal here is not just because of spiking activity. There's something else that contributes to it, and we have to be very careful to try to understand this part. So let's get to it. Okay, so again, a quick review. This is how a neuron looks like, right? So we did this in the previous class. These are the functional regions. And if you remember that one neuron is connected to another neuron through this, what is called a synapse. This is the axon of the presynaptic neuron. So this is the neuron, let's say, from LGN, and this is the postsynaptic neuron. This is the V1 neuron, let's say. So this guy is going to release some neurotransmitters which are going to bind to the receptors on the other neuron, dendrite of the other neuron. And this is going to lead to opening of some ion channels, and some ions are going to flow in, which will depolarize the next neuron. We have done all of this, right? Here is a sort of zoomed in version of the same thing, the synapse here. Action potential reaches at this end. It causes these synaptic vesicles to fuse. The neurotransmitters get released. They bind to the next neuron. And every time this, this is like a bunch of neurotransmitters that are releasing, and there is a slight depolarization because of the opening of some ion channels. This is the intracellular potential of the postsynaptic neuron. And if enough packets arrive, so enough action potentials and enough of these vesicles are released, then it's going to fire an action potential. This is the postsynaptic neuron firing an action potential. So these are the synaptic events. This is the action potential. These are two different things. It is possible that neurotransmitters are released, but not enough are released. So this neuron doesn't end up firing an action potential. Once the the cell gets depolarized, there's some leakage, remember, from the passive channels. So unless more of them arrive, it can come back and never lead to an action potential. That's the logic, that many neurons have to fire together, and the synchrony matters. Arrival of these synaptic, these packets matters, and so on. So enough of them have to arrive for the postsynaptic neuron to fire. And there is a chance that the synaptic activity nonetheless doesn't lead to an action potential in the postsynaptic neuron. Everybody happy with this? This is fine, right? We covered all of this in the previous module.
Yes, yes.
Okay. So now here's the deal. Actually, let me just show you this on. So I will then come back. I'm just going to describe this. But let me just draw this on a Jamboard here. I used to do that on a blackboard, but this is online today. So suppose this is a postsynaptic neuron. So it's got this little dendrite here, and this is the presynaptic guy, presynaptic. So this guy releases neurotransmitters. They bind here. What happens? Okay, so I have got an electrode here. This is an extracellular electrode. Okay, tell me something. Suppose I've got an electrode close here, and this guy fires an action potential, releases neurotransmitters. What's happening right here? What would you see as far as ions are concerned right here? Anyone?
Movement of ions.
Movement which way?
Because of these neurotransmitters would let the ion channels to open, and then ions will move. Some ions will move inside the postsynaptic neuron.
So initially, the sodium channels will open, right? So sodium current is going to go in, and that's going to depolarize this guy, right? Now, if I look at this from outside, suddenly I will see that the positive charge here got, it just sank, it just went in, it just disappeared, right? So from the outside, this is going to be like a current sink. This is a current sink. Suddenly there was, charge here, and like a straw, somebody just sucked it. Zoop, it went somewhere. Where did it go? Inside the cell. But here, all the positive charge, the sodium that was there, suddenly just got sucked somewhere else. So this is a current sink. Now, what happens to this charge? So what happens to this charge?
It becomes more negative than the previous one.
No. Well, so charge basically has gone in the cell, positive charge. What will happen to that charge?
It will make gated potential.
Graded potential. Okay, no, that's true. So inside the cell, if I look at the potential, it will be depolarized. So the potential Vm inside, indeed, from -60, it will start to increase. You're right. That's the same as this other plot. I was showing this thing here. I'm not talking about the potential. I'm asking about the charge. This positive charge here, the extra sodium, what happens to it?
So after a while, it might pump back outside.
From where?
From this cell to the extracellular region.
Okay.
Pumps come to work here? Because I mean, if it's not creating an action potential, there is not going to be sodium ions. Like I mean, even if there wasn't an action potential, the whole point is to maintain that equilibrium, resting state potential. Those sodium ions eventually have to be sent out with the pumps. So those are not going to hang around for too long, is what I'm thinking.
Yeah. So in the long run, yes, but actually, the charges that you have, it's very hard for you to hold charge. It tends to get balanced out. Okay. So actually what happens is that the charge that comes in actually flows along the length of this axon, or here it's not the axon, it's the dendrite, and it tends to flow out from other regions. This is actually why the rest of the membrane is also getting depolarized. Okay, so the charge won't hang around here, or for that matter, wait for the pump to go back. It's actually an axial flow of current, and it actually goes out from somewhere else. And this is not after a while. This happens more or less instantaneously. The net current, the net sum of transmembrane current is actually zero. This current, actually the transmembrane current, which is the current that flows in and out of a cell, goes through two routes. One is that it goes through the ion channels, which is the ionic current. And the second is that it gets deposited along this, which is essentially constituting the capacitive current. Remember this ionic and capacitive current? We did that in the previous module, where we looked at the currents that are going in, and there was a capacitive component and an ionic component. But that total current, which is the sum of the ionic and capacitive current, has to be balanced out so that the net current, net transmembrane current, is 0. Okay, and these actually constitute what are called distributed. I can't write here very well, so I'll just put it. These are distributed current sources. So the extracellular, in the extracellular world, what you see is a separation of charge. A separation of sinks and sources. If the cell is very small, the sink and source are going to be very close to each other. And then it won't be much, actually. It won't constitute too much as far as the extracellular potential outside is concerned. But if the cell is big enough and the sources and sinks are separated, this together, what is called, if it is just a minus and a plus here, this is what is going to form what is called a dipole. It's the separation of charge, minus and plus, along some axis. And these dipoles produce these kind of extracellular potentials. Okay, so if it's a very small spherical cell, it actually won't produce much of extracellular potential, because these charges in and out will be very close to each other. But for larger cells, which are long here, like pyramidal cells that we have, the incoming current and the outgoing current are separated, producing small current sinks and sources, which can produce these dipoles or even higher order poles. If you have a minus here of whatever charge P, and a plus here and a plus there, plus P by 2 and P by 2 here. This is a higher order pole. This is a multipole. And there the potential falls off in a different way as you move away from this multipole. Okay, so this is fairly physics heavy stuff. So what I told you, this is again taken from this review paper, but it's well accepted. So let me just read this out. Extracellular currents from many individual compartments must overlap in time to induce a measurable extracellular signal. Okay, so synaptic currents are actually good candidates because they are relatively slow events. But the stuff that I told you is this: the influx of cations from the extracellular to the intracellular space gives rise to a local extracellular sink. This is the current that got sucked in. However, to achieve effective electroneutrality, the extracellular sink needs to be balanced by an extracellular source, which is an opposite ionic flux from the intracellular to the extracellular space somewhere along the neuron. And then depending on the location of the sink and the source, a dipole or a higher order n-pole is formed. And it is these dipoles that constitute your extracellular potential. Okay, and I will end by just telling you the formula for it. This is again, there's more physics here that we will handle as of now. But when we come to the EEG part, I will actually dig into this one more time. At the time, you can get a better feel of this. This comes directly from Coulomb's law and the Faraday's laws. Not deriving any of this, but here's the setup, the whole picture for you guys. These are pyramidal cells. They're all lined up like this. In each neuron, you can divide it into small segments and then talk about the transmembrane currents, the currents that are flowing in and out of the cell. And here's my extracellular electrode, the positioned here at this point, and I'm measuring the potential here. Oops. With respect to some ground reference electrode, which is farther away, quite far away. And I'm looking at this potential, the contribution of this potential because of this little current here. Okay, and this formula tells you what it is. For the nth current source, so I've divided into n compartments, okay? Each neuron into n compartments. I am looking at the contribution of this current here on the potential that I record here, and it's nothing but the current divided by the distance Re minus Rn. So this is the distance here divided by this 4 pi, and this is the conductivity of the extracellular medium. And then each of these currents are going to contribute a single term, and the total potential is for that one, from that one neuron, is the sum of all these. Notice if the neuron was very, very small. Oh, by the way, so if you forget the denominator, summation of I and t, I going from 1 to n, what is that? The total net transmembrane current. What is that? Based on what? 0. Very good. Based on what I told you, the total current flowing through the membrane is actually 0. The total sum of the capacitive and ionic current is 0. But the currents are going in and out from different regions. So the denominator comes into the picture. Because the denominators are different, that is why the sum here is non-zero. For this to happen, you need a long cell. The pyramidal cells actually contribute more extracellular potential than, let's say, the inhibitory interneurons that are smaller in size, because their currents are all very close to each other. But in a pyramidal cell, the sinks and the sources are separated, and this formula tells you the potential of a single cell. And if you've got like 10,000 cells or 1,000,000 cells around this electrode, each cell is now going to contribute a potential, and this is the overall field potential. This is what I see here, the raw signal. Okay, and it's very important to realize that if none of the neurons around this electrode are firing action potentials, if none of these neurons fire action potentials, that doesn't mean that this potential is going to be 0. Because this potential is because of synaptic activity, the opening of ion channels. Even if they fail to elicit an action potential, nonetheless, whenever ion channels open and current goes through, sources and sinks are going to be produced. So you will see a potential here, even if you prevent all these neurons from firing action potentials. As long as the previous neurons that these neurons are connected to are firing action potentials and causing these ion channels to open and close, you will see a potential here. So this is because of synaptic activity, not necessarily only because of action potentials. Okay, and I'll let that thought sink through and continue from here in the next class. So please think about this. The LFP that I'm going to get to, this is just the overall field potential, okay, the raw signal. It constitutes a lot of things, including the contribution from action potential, but it's the synaptic activity that dominates the show. So we are looking at the opening and closing of ion channels and the currents that are flowing in and out of the cell that contributes to the potential that I record according to this formula. Okay, so I will stop now, stop recording, stop sharing. You are free to go to your next class, and I will